What's your favorite day at work? Like the day you dream
New
of?
data day. Sorry.
What's your favorite day? What was it?
New data day.
New day today. You have a new day today? When's the new day today?
No, any day can be new data day, but it's so much fun. Like when you get. Is that what you're asking, or is it like just a Tuesday?
No, that was a good answer. I just
Okay.
never heard new day today said before. I mean, that's really wonderful. Hello, everybody. This is the Dairy Digressions podcast. I am Matt Lucy, and I will be your host. We are bringing this special episode to you from the 50th Annual Conference of the International Embryo Transfer Society being held in Denver, Colorado. I'd like to thank the IETS for their willingness to have us interview some of their invited speakers. Today, we are speaking with Professor Niamh Forde, Professor and Chair of Molecular Reproductive Biosciences based in the School of Medicine at the University of Leeds. She is co-founder, co-director, and academic lead for LeedsOmics, a virtual research institute. She's going to tell us about her research on embryo endometrium interactions in the bovine. The Dairy Digressions podcast is where we talk to some of the most innovative minds in the field of dairy science. Dairy Digressions is brought to you by the American Dairy Science Association, the largest dairy research science society in the world, fostering communication and collaboration through its journals and meetings. As we will find out in the podcast, we have with us an Irish lady, and I found out one thing on my sabbatical in Ireland is the Irish don't go to cold places for vacation.
No.
Why would you go to someplace cold and wet on vacation?
Because you live there all the time.
All the time, right? So anyways, if you enjoy Dairy Digressions, please listen, like, and subscribe. And if you have feedback or suggestions for speakers, please tell us at adsa@adsa.org. On with the podcast.
Awesome.
Welcome, Niamh.
Thanks, Matt.
So good to have you here.
Thanks. It's great to be here.
I don't know how we know each other.
Nor do I, actually.
I don't really know.
Yeah, I don't know.
I mean, there's only 4 million Irish. I know most of them. Okay, so maybe there's 5 million Irish. I don't know. No, 5 million Irish, 1.4 million cows in Ireland, and 30 million Irish in America. So just remember that statistic, okay?
Definitely will.
Yeah, Joe Biden is Irish, I think, right?
He is, yeah, but his family are from Mayo, like a competing county of ours.
Oh,
so,
no.
you know, he's all right, but he's not like, I joke.
So we always go through these counties in Ireland, okay?
Very
It's a
planet.
big deal, all right? I'll go through it. So you're born in Ireland, what county?
Galway, the best
No
one.
way, Galway?
Yes, I grew up in the town centre down from Earth Square. Yeah.
Are you from a farm there, or?
Nope.
You were born in Galway?
Yeah.
That is fantastic.
Yeah.
I love that city.
Yeah, no, it's great, but I was concerned when people visited it for the Ruminant Reproduction Symposium, because we had such wonderful weather. I had to go around telling people at the conference that this is not what it's normally like. This is like a once in every 4-year event. It's lovely, even when the weather isn't delightful.
Yeah, so the Ruminant Reproduction Conference, which is held once every 4 years, hosted by the Irish this year, did a phenomenal job. Sunshine every day, swimming in the ocean every day. I mean, Ireland is a lovely country. Now, this really surprised me. You can explain the significance of this.
Okay.
Did your undergraduate at Trinity and then went to UCD?
Yes.
Now, that's broken, isn't it?
Not really. So I would say...
Not really broken? So it's a little bit broken. Okay, for everybody, Trinity is the big bad school in downtown Dublin. Okay, fair enough.
Yeah, it's a great university.
It's got a great green.
It's my alma mater. I have to say nice things
Oh
about
yeah,
it.
Trinity. Okay, okay, great university.
Yes.
You majored in biochemistry.
Yes, I did. Yeah.
Why in the world, I mean, in Galway, you grew up and you said to your Mom, Dad, I want to be a biochemist.
I think you give me more credit than I deserve. I went to university when I was 16, but there was a perfectly great uni 10-minute walk from my parents' house, but I decided to go across the country, and I picked, well, actually it was between science and history and politics.
I
to
love
do my
it.
undergrad. So I guess I've probably, and it's maybe a good thing to be an academic, but I've always been a bit like, if I like something, and I'm really interested in it, I'll be very enthused about it. But if I don't like something, I'm not very good being diligent.
So do you think when you went to Trinity, was it a heavy lift? You go in at 16, that's a pretty hardcore school, right? Trinity is very good
but
in
I
the
was
basics.
also like a terrible undergrad as well. Like if I had had me as an undergrad now, I'd be like, you're really, please just try a bit, you know? So I wasn't great, but I eventually kind of found my feet kind of towards the end of my degree. And actually, when I was in my last year and I was taught a bit of repro, I tried to purify an acidic glycoprotein from a mouse epididymis as my final year project. And I was like, oh, this is it. This is the cool thing that I want to do. This like actual proper science, if that makes sense.
Right,
So I don't think it was actually, up until that point, it wasn't, oh yes, I'm going to do this thing. Because no one in my family from my parents' generation, my parents didn't go to university or anything or finish high school. My mum in particular was always like, education is really important. So there was no barrier for us being able to go. But I don't necessarily think I did anything on purpose up until maybe applying for like a PhD.
I'm older than you. My mother, she didn't go to college. She was just told girls don't go to college. And my father went to night school, but it was a very different, there was this weird expectation in our family that everybody would go to college. Kind of interesting, you know, and so it was a very different time. You know, my parents had no experience with college. They knew it's what you needed to do.
Yeah, and I think maybe that's probably a bit similar to the way my parents were, and my mum in particular was, I didn't necessarily have this opportunity, but absolutely you all are going to have the same opportunity and go for it. Maybe that's a common theme.
And then off you went, huh?
Yeah, I did indeed.
You did, and then you
stayed
found
in universities.
You found the mouse epididymis, and then you were like, oh, now I'm going to go to UCD because... Okay, University College Dublin. Okay, excuse me. So Trinity's a beautiful school downtown. If you go to Dublin, go to Trinity, walk around the green, spend a nice afternoon in the sun, lounging on the green, watching the people go down. It is beautiful. Okay, UCD is a little outside of town in Belfield, Belfield. Okay, so you got to take a bus. It's probably number 67 bus. I don't know. There's a bus to there, but it's very different.
Yeah, it is. But that's where I got the opportunity to do a PhD. So,
with Alex Evans?
Yeah, on follicle development, and it was kind of my first introduction to cattle as well. Yeah.
Is that right?
Yeah. It's like baptism of fire in terms of doing animal work. So growing up, the largest pet we had was a cross between a terrier and a Pomeranian dog. So working on beef cattle then was a slightly different scenario. I had to learn pretty quick.
Is that right? So Alex Evans, big shout out to Alex. I think he's dean of the college right now, or maybe he has retired.
He's just finished that position, as dean.
Yeah, and of course Alex, he's Irish, yes.
Yep.
And of course he transitioned through Cornell at one point with Joanne Fortune, who's another friend
Saskatoon.
And then friends of the American Dairy Science Association, UCD. Was Jim Roach there when you were there?
So Jim Roach was my first boss when I did my postdoc.
No kidding.
Yeah, so I didn't work in anything to do with the uterus so I finished my PhD with Alex and applied for a postdoc with Jim Roach, and the project was on looking at how progesterone affects the endometrial function in cows. So that started me off on my uterine biology adventure.
So I think it's important to note here that not everybody will know this, but these, when we throw around names like Alex Evans, Jim Roach, iconic individuals when we talk about bovine reproduction, the biggest of the big names, okay? You must feel fortunate to have trained with these individuals.
Absolutely. Very robust experimentalist and experimental design. And I definitely think I was fortunate to get the thought process of how to think like a reviewer almost when you're designing your own experiments, so being your own critic as well.
Yes.
And I think that was definitely something that served me well.
And so the Lonergan thing and with Trudy, when did that all start? Was that a postdoc or what was that when you started working with Pat?
So I started working with Jim in the first instance as a postdoc, and then Jim kind of retired. Nothing to do with me, I would like to add. He was coming towards the end of his career and kind of was transitioning towards retirement. So I was fortunate enough to be able to get some experience of managing grants before I had to really do them myself. And then after he kind of retired, hired and that project finished, I then began to work with Pat as like a research fellow, again looking at early conceptus maternal interactions, and then doing a large dairy cow experiment, looking at lactating and non-lactating dairy cows and lots of different points on the reproductive cycle to try and understand what components, how lactation affects embryonic genome activation, pregnancy recognition, lots of different things like that.
Did you ever grow to like the cow, or was it just you tolerated the cow, or was it?
I mean, I think they're delicious.
You like to eat the cow?
And they make delicious products. I love a cheese board. No, I do really like cows, and I used to have, I know you shouldn't say it, but I used to have favorite ones when we'd be doing or experiments and stuff. Yeah, no, I do like cows. I think they're lovely animals.
Yeah.
So I do, yeah.
Because Irish, everybody in Ireland owns a cow, or their grandmother, somebody in the family owns a cow.
My grandfather had like a small farm.
Yeah, there you go.
Yeah, I think way back when.
So here we are, you're finishing up. What was the thing that tripped in your mind that made you think, I need to get out of here, start my own lab? When did you, because you'd been at UCD a long time.
Yeah, no, I had, I guess, a couple of things. I had wanted to stay in Ireland, and then just for reasons, felt like it was now a time that I could move. And Ireland is very small in terms of the number of opportunities to get like a tenure track position, because there's not that many universities there. It kind of all culminated in me being like, okay, I think I need to either now decide that I'm going to go to get a PI job, or accept I won't be able to probably stay in science. So that's why I began to look further afield. So while I was still at UCD, though, I had been applying for jobs but in Ireland, but never got. But in hindsight, that was actually good, because you know yourself, moving around, getting to talk to new people about science and collaborating with new people always sparks like new ideas. So sometimes you have to be kind of forced to do it from circumstances that you mightn't necessarily have wanted, but yeah, I think sometimes it's good.
I'm like you, I had a very nice lineage. I worked with some really excellent people, you know, and it really set you up, and then moved around quite a bit. And you're right, every time you move, you meet new people and make new connections. And so Leeds came up, off you go to Leeds. You're like, well, I'm gonna have to find this on the map first, probably. I mean, does everybody know that there's a university in Leeds? The only reason I know it's there is because of you. Yeah. They call it the Little Oxford, right? Or
sure, no. So I guess it mightn't be one of the better known universities. So in the UK, everyone knows like Oxford, Cambridge, and then there's like a group of what they call the Russell Group universities. So a bit like they're called the Red Bricks, like Manchester, Leeds. I should know all the names of them, but I can't remember right now. I'm going to blame jet lag. So I did know where Leeds was, etc., but maybe not everybody knows. And there's a few people there doing some repro stuff, but it wouldn't be like as well known as the Rosalind Institute in Edinburgh, or some other kind of hotbeds of reproductive greatness, as I call it. A bit like Missouri. So maybe not so much like that. But I think one of the benefits to that is it forces you to interact with people and think differently about the science you want to do and how you do it.
So there's a medical school there.
Yes.
So there's got to be a finite number of medical schools in England, for example. So that makes it one of these brown universities, the fact that there's a medical school there? No,
there's an awful lot of med schools in the UK. So it's not like maybe the US so much. There's more limited vet schools in the UK, I would say. But the reason I was in the med school is because the colleagues working in reproductive biology were based in the med school, even though like colleagues use lots of large animal models and bovine and ovine and stuff as their model systems as well.
What year was that you moved from UCD to Leeds?
In September 2015.
So you had about 5 years until Brexit hit, and 5 years until COVID hit. So you're probably trying to make tenure or something like that. Brexit was a real shock, right? Because that changed the landscape for grant funding.
I'll try to remain calm when I talk about Brexit. I won't succeed, but I'll try a little bit. So one of the so many unfortunate things, but if we just focus on funding for science, is the fact that there was an awful lot of uncertainty as to whether or not the UK, and if you're a scientist in the UK, that you could apply for EU funding, so what was Horizon funding or ERC funding. And it took an awful lot of negotiation, and basically a couple of weeks ago they signed.
I read that, yeah. They finally agreed to it.
I mean, the government were like, woohoo, look, we're accessing all of these things. But actually, we're in a worse position than we were before Brexit because you don't get all of the benefits. But anyway, at least people can apply for funding again. I think psychologically, it's done a massive amount of damage to the reputation and for people's willingness to move there as well. I think in terms of science, like even trying to hire people who aren't from within the UK, the visa processes are much more difficult. And I mean, I'm fortunate I'm Irish, and because of our interesting history with the UK, I didn't have to apply for settled status, but every single other European person who had been working for years in the UK had to apply for the ability to stay there after Brexit happened.
What?
Yeah, there's so many unfortunate things.
It was a big deal.
It was, and it still is, to be honest. And I think particularly being an Irish person there, it made me very aware of maybe how little people understand about the history between Ireland and the UK.
I know all about it because I spent a year in Ireland.
Yeah.
You know, and that brings up a funny story. Would you like to hear a funny story?
I'd love to hear a funny story. I'll tell you mine off mic.
When you spend any time in Ireland, you quickly learn that there's something with the English the Irish don't particularly like, okay? And then a minimal amount of studying, you start to understand there's a lot of history between the two countries. Fair enough?
Yes, I think.
And it goes. It's a deep history.
It is
It's
indeed.
uncelebrated past.
Tumultuous, I would call it.
Tumultuous, yeah. This is a terrible story.
Okay.
Terribly embarrassing for Dr. Lucy, okay?
You're talking about yourself in the third person, so I can imagine it's terrible already.
So there I am, and I'm at this conference, and there's Dr. Roach.
Jim Roach?
Jim Roach,
Okay.
the man. I had read all his papers. He's so famous. And I just wanted to go up and say hi. I said to him, hello, Dr. Roach. My name's Matt Lucy. How's everything in the UK? And he looked at me and said, everything's fine in Ireland. Thank you very much. Okay, so at that point,
I'm sorry.
I went back to my friend. They're going to cut this all out of the podcast, so don't worry.
Okay, that's all right.
So
Good.
I go back to, they're definitely going to cut this next part out. I go back to my friend, John Byatt, who I was working with at Monsanto, and he's an English guy.
Yeah.
And I said, John, I explained to him what happened, and he said, can you explain to me the difference between Ireland, the UK, and Great Britain? And I'll never forget. So he goes through it, and then he finally gets to Great Britain, and he says, we used to call it Great Britain, but we're not all that great anymore, okay? So anyways, I thought to myself, okay, now I understand. So anyways, he was very gracious, okay? But it was so embarrassing. Such an embarrassing thing to say. So then COVID hits. Did you shut down your lab for how long in COVID?
It was really hard. It took at least a year for us to get full access back to our lab.
Okay.
We shut down in March. No one was allowed in. By September, a couple of people were allowed in, 50% of the time, but on it almost like an emergency basis. So these are people who had to finish their PhDs and stuff. And then it was part-time, 50% till 2021, kind of the mid of 2021. So it really, really impacted in the lab. I wasn't allowed in. Everything was done online. And I think not just the not being able to be in the lab or do the experiments or whatever, but a lot of the science learning that you would do accidentally, you'd be someone would pop into the office and students would come in and you'd end up chatting about stuff or just wandering around the lab. That didn't happen, and I just feel really bad for the students and postdocs who were working at that time because they didn't get the experience that you need and the science interaction and the fun, and they didn't get any of the fun bits. We have such a great job, right? We get to go to Denver, Colorado in January and sit here and talk to you about a podcast, but there's all these, science is wonderful, but the job is difficult. If you're a grad student, postdoc, pre-tenure, tenured, there's difficult bits at different stages, I think. And one of the most wonderful things about what we do is being able to talk to other people about science and do that in wonderful places. And I think not being able to do that has, I think, been quite detrimental to trying to keep people in science a little bit at the moment. And I just felt really bad for students.
Yeah, it's the best part of it all, right? We always dread getting on airplanes and flying to different places, but when you get there, it just kind of gets you going, and you sit through different seminars, and you're like, oh, I didn't expect to get this here, but this is decent, and this is important to me. So you made it through, because I know you were trying to get promoted or whatever happens over there. I don't know.
Yeah, not get fired.
Not get fired. You're trying to get not get fired. You make it through there.
Equivalent to tenure, yeah.
I have a question. How many people in your lab right now? You show all these pictures.
Right
How many
now,
people?
so right now, loads of people have finished. So we currently have 3 people in the group, and we're just hiring 4 people.
3 plus 4.
Yeah,
7.
which is a nice number of humans. At one
Okay.
point, there was like 14 or 15, and I think that's too many.
That's
for
too
me
many.
To manage
Yeah.
anyway.
What do you think your mentoring style is? Do women have a different mentoring style than men? Do Irish have a different mentoring style than Americans?
I think
All
you'd
those
have
questions.
to ask the people in my lab. So I had this very unrealistic thought before I started my own lab, which is like, I'm going to treat everybody the same.
Oh, God.
Which
yeah.
is like the worst thing you can do, right?
Right.
So like I figured that out very, very quickly. I feel like everybody's an adult, right? So I'm not going to micromanage people. I don't micromanage people, and just give them the tools to do the good stuff that they want to do. I like want to be supportive and give them opportunities depending on what they want to do. I'm not sure I have a mentoring style, if that makes sense, because I don't think you can have just one way to do it. Because if you do, you treat everybody the same, and that doesn't get the best out of the people that you work with, I think. But definitely, I hope not a scary type of mentor.
Yeah.
Like, you know, the way there's certain people who be like quite direct.
Yeah.
Yeah, I don't think I'm like that.
Yeah. The Irish are generally not all that scary.
I don't think so. Yeah, no, I don't think so. I think we're quite,
Yeah,
just
you're
pat
not-
ourselves on the backs. Yeah, we're all great or whatever.
Yeah,
but
they like to get along and
like a collegiate approach to working rather than like not a fearful approach, if that makes sense. I hope.
I can understand that. I'm also very hands-off mentor because as soon as you start telling people what to do, they'll just be asked to be told what to do, you know, and you know, you try to give them objectives and this is what we're trying to get done here. Yeah. And you know, one thing what's very fascinating, you probably had the same experience, is when you start out your lab, you have this assumption that everybody would be just like me, you know, and then they'll just be like me, me and Dr. Thatcher, here we go, okay, it's gonna be great. Yeah. And then you quite realize that everyone is different and everyone brings different things to the table, and you just have to work with that people are different.
I do try to be honest with people, and I know that sounds like a very obvious thing, and honest with the people in my lab as to what I'm doing. And I always say to people when like I'm meeting with them or having an update, I'm like, look, it's not that I don't care about your project, it's just that's your project, right, to you, whereas I have X number of them, and you just need to remind me and then I'll remember and stuff. But being honest with them so that they understand the number of different plates you're spinning at the same time and things like that.
Yeah.
Because I think sometimes difficulties can be born out of frustration as well, without people not necessarily understanding. Obviously to you, your paper, your thesis chapter is the most important and urgent thing to get done, whereas explaining that like there's all of these things and putting all the bits of the Tetris together, I think maybe helps.
What's your favorite day at work? Like the day you dream
New data day. Sorry.
What's your favorite day? What was it?
New data day.
New data day. You have a new data day? When's the new data day?
No, any day can be new data day, but it's so much fun. Like when you get. Is that what you're asking, or is it like just a Tuesday?
No, that was a good answer. I just never heard new data day said before. I mean, that's really wonderful. Okay.
Yeah, but that's what we do. Like, and then being able to get excited and talk to the people in your lab about it and what it might mean, and that's the best bit of the job. And then obviously when the people in your lab do really well as well, that's really good.
I do love new data, and I love to receive. We were talking earlier with Carl Zhang from Florida, and it's funny, he just brought up, I said, what's your favorite day? Oh, Friday is my favorite day. I said, why? 9 a.m. lab meetings. I love lab meetings, you know, because they have all this data, you know, and so I love snow days too, but you probably don't get it. Do you get any snow there?
Yeah, sometimes, yeah.
I love snow days because it's generally really quiet at work, and you can sort of work away and, you know, and get some stuff done. And that snow days are great. I love snow days. But, you know, that's what makes working really special, right, is new information and seeing these kids sort of, you know, develop and get good at stuff and go off. So what the heck is LeedsOmics? What is a virtual science? I mean,
Research. It basically means that, so when we started at Leeds, there was a big recruitment drive, and a couple of, like me and two colleagues now, my closest friends, were like, hey, we use omics, but where are the omics people here? And there wasn't like any community, so we sort of formed it ourselves. And we got to hire some people to do bioinformatics support for not just us, but other people who needed that or had never done any omics analyses before. And we've been involved in like training over like a couple of hundred students, postdocs, like PIs over the last 6 years or so. Just kind of really about community building, because that's the thing. I think that for me is one of the things I love about like science and the job we do, right? Is you get to talk to people who like are as nerdy about science as you are, and I think it can be quite a lonely job sometimes. And you know, as I like to say, I get professionally rejected like all of the time, and so you need to have your community to, like we get critiqued and papers, grants, everything, talks, and that's part of the job. But you need your community of support to be able to help you navigate that, I think. And if you don't have that community, then I think you need to make it.
Yeah, I think that's important, right? And I think one of the things, as you'll agree with, is that science has really changed, and so you need a lot of people to get this stuff done. You can't possibly have the expertise in your own little head to deal with all this stuff anymore, and you know, you need to draw in different people and a little bit of trust maybe, and there's good collaborators and bad collaborators, and over time you'd kind of stop working with the bad collaborators and you work more with the good collaborators. How's that sound?
I think you have to be quite open to collaborations and interacting with new people. Like I do like to have a bit of a collaboration test, which is like, is this somebody who I will happily have a coffee with or go for a beer with, and could you maybe have a difficult conversation with them if you need to as well? And if you can, I guess maybe the having a coffee or a drink with them is like, can you communicate effectively with those people? And I think that's probably what collaboration is about, learning each other's languages and how you speak and how you get on with things.
You've really been successful at that. I was looking at your publication list, and you got lots of different people and that you work with.
I'm very lucky.
Yeah, they're fantastic. I want to ask you about a couple of things you really kind of brought up. So we're at the IETS meetings, and you gave a talk earlier, you know, when you were talking about some of your science, your love of the endometrium, which we're going to talk about in a second. You also sort of hinted on, which was really interesting, placental development, which, as we mentioned before we started the podcast, if you take something like a heart, for example, and if I put a heart of a cow, a dog, a person, a mouse, I don't know about a possum, maybe. I never looked at a possum heart.
I'm sure.
I put it on the table, it all looks pretty much the same.
Yeah.
They're all the same. Got 4 chambers, very big aorta, vena cava, the lungs get hooked up to them somehow, right? It's all kind of the same biology.
Yep.
But if I put placenta from a human, a dog, a pig, a cow on that table, it's all over the map.
Yes. And why wouldn't you want to understand why that happened?
Well, why wouldn't it be the same? That's stupid. Why did biology come up with this stupidness, okay?
Have you seen a marsupial? Oh my
They're
God.
messed up. I don't look at them. They're scary. They have teeth.
Amazing.
They're amazing,
The diversity in biology, species, placenta, whatever, is just fascinating.
So is this sort of a curiosity of yours, or would you call this a main research focus to understand the diversity of placental types? Because let me point this out to you, Niamh.
What, Matt Lucy?
I mean, there are people that spend their entire lives just to understand the development within the heart, which is all the same.
Yeah.
And now you want to study the placenta, which is completely crazy. How?
Why not?
Why not? You're just curious.
Yeah, isn't that the job?
It's bizarre, right?
Sorry, I don't know. I don't know the correct answer.
There is no correct answer. You're back in your comprehensive exams, okay?
So it's absolutely fascinating to me that you have an end point, which is a successful offspring. So if you're a dairy farmer, or you work in conservation biology, or you work in assisted reproduction in humans, what you want is like a healthy offspring. Can I call a human an offspring? Yeah, we'll go with it. Anyway, but the way to get there is so diverse. It's wonderful, but like how is that happening? And what are the things that are conserved among them? Like, so what are the really important things that happened to allow placental mammals to be here? And then what's allowing this wonderful diversity in these organs and how it interacts with the maternal side. Like, yeah, what's driving that diversity?
It's fascinating, right?
It's incredible.
Pregnancy itself is fascinating.
It is. I get on my high horse a little bit about this sometimes. So many people work in other systems, cardiovascular, whatever,
Yeah.
and it seems like pregnancy in domestic species, which is super important for food, wild species for conservation and humans, and something that establishes life course health for individuals and is so fundamental, is so poorly funded or recognised.
Right, yeah, fair
And
enough.
it really annoys me. Yeah, and I think we need to be better at advocating for why what we do is so important. And I'm sure, like, I know every scientist thinks what their thing is like the coolest thing or the most important thing,
Yeah.
right? But actually, I think we need to be better at getting everybody else to recognise why what we do is so important.
I think your fascination with the placenta is really a fascination with developmental biology and sort of over time and how these different structures have evolved over time in the development
But
of
how
that.
the uterus supports that, because
Right.
actually the uterus is the coolest organ, because
Right.
it's the thing that's allowing the support and growth and development in a super diverse manner, right?
Yeah, that's always going to be good, like developmental biology or how these things work
Yeah.
and how cells work
Yep.
is always going to be good. I was listening to Stem Cell Podcast. Hey, Daylon and Arun, someday they'll come to my podcast, okay? On the way here, you listen to Stem Cell Podcast?
No.
No, okay, yeah, okay. Well, you need to listen to the Stem Cell Podcast, okay?
Okay,
And
yeah.
they were just talking about neuronal development and how, you know, in a mouse brain, it develops in a relatively short period of time, and a human brain develops in a really long period of time. And they were just mentioning how, you know, if you take immature neurons out of a mouse brain and put them in a dish, they develop really quickly. And if you put immature neurons in a human brain and develop in a dish, they develop really slowly, okay? And so it's fascinating, right? So these cells have this sort of intrinsic
Programming
sort
sort
of
of
programming, and so it's really interesting. And I know you're working on microRNAs. MicroRNAs are super hard, but I think your general thought here is that the microRNA population has something to do with sort of the invasiveness of the trophoblast. Is that fair?
Yeah, I think because they seem to regulate lots of different pathways and things, I think that they're at least one part of the puzzle, right? So science is great. We'll never run out of a job because there's always something else to look at. But I think they probably do form an important component of it because they regulate lots of different pathways. Was
Yeah.
that the answer to the question?
Yeah, fair enough. Yeah, good.
And I think if you think about having to establish a new system, so how the embryo and the uterus interact, changing microRNAs might be a good way to do it without having to change loads and loads of pathways.
Yeah,
right?
fair enough, right?
Yeah, it's a bit simplistic, I suppose, but yeah.
One of the things I thought's been really interesting about your career is sort of first of all, your fascination with endometrium, your fascination with sort of developing more advanced models for studying endometrial function. And the paper you submitted to this conference was really kind of interesting because there's a big section on it. It starts with, you know, you're talking about, okay, two-dimensional, basically growing cells on plastic, growing cells on matrix, and then trying to understand their function. And then finally, you also talked about explant cultures and their strengths and weaknesses, and then made your way into sort of organoid models and that sort of thing. And of course, organoids are pretty hot right now, and also microfluidics are pretty
Yeah.
hot right now. And
I know it's like hipster science bingo.
so
Yeah.
let me ask you where you are on organoids. We'll stick sort of the endometrial luminal epithelial cell type organoid out there. Where are we in terms of organoids into sticking a stromal cell layer beneath that or into it or something like that? Where does the science start right now? Okay, so for the Stem Cell Podcast listeners, we have got these luminal epithelial cells, and they're super cool, sort of columnar epithelia lining the uterus. But everybody knows that.
The glands are where it's at, right?
No, well, you start with the stroma. So where are we at with putting those two bad boys together? They're like brother and sister.
So we have in the lab been working and are working at the moment on having stroma that deposit their own extracellular matrix, and we have managed to layer on luminal epithelium
I
onto
saw
those.
that. Looked pretty nice.
What we're working on right now is trying to see when we can potentially drop some glandular organoids into that system.
A glandular organoid.
Yeah.
So that's different from an organoid or what?
No, but like that they're more glandular in their phenotype rather than luminal epithelium.
Okay.
Yeah, so that's one of the things we're working on at the moment. I would like to preface all this by saying I think the best model to test your hypothesis in is the in vivo model that you want to work in, so like cows, right? But I think there's lots of things we can't do from ethics perspective.
You work in a medical school, you also can't do that in women, right?
Exactly.
So, okay, I'm just saying, I mean,
no, but that's what I mean. I think
I just bring up, I can bring up a point here, cows or women, so you have to have these models.
Exactly, and so I think, hopefully I got it across in the paper in my talk, is that I'm very aware of the fact that an in vitro endometrium is not equivalent to an in vivo cow scenario or a human. But we also have to understand the limitations of all of the model systems we're working in, and I think maybe one of the things that tends to happen is people are like, I have the best model, and this is the only one that will work, and actually we need lots of models and lots of different systems and lots of people working on them. So we're able to answer different questions.
You sort of seed these stromal cells into some sort of matrix, and they're laying down extracellular matrix for you, and then lay those other cells on top, right? But now you got to get some cells that'll basically invade and make glands. That's a heavy lift, right?
Yeah, so we're trying to do that drop-in of the glandular organoids as the stroma is established in the extracellular matrix, but they're super long experiments and there's a lot of difficulty in terms of what the different cells require, etc. So we have a lot of troubleshooting to do on them. But we're fortunate at the moment that we have some funding to look at that in a variety of species. So in humans, in cow, and also some pig models as well. So hopefully we'll be able to learn as we develop them from species to species as well and make things a little bit quicker.
And then the second thing I want to ask you about is your microfluidics, which also look pretty decent. So you've got sort of these microfluidics. Who are you working with? Engineering department on that or what?
Yeah, so the one I presented today and the one we published, which we tried to mimic glucose and insulin in negative energy balance, dairy cows, is just a commercially available one, but we work with a bioengineer in Leeds, Virginia, and she can fabricate one. So she's fabricated some that we could add these glandular organoids in and try and put them under flow. So we're very fortunate to have those kind of colleagues that are happy to collaborate.
So do you think this 3D printing of tissue is ever going to happen? Or some people are trying it right now where maybe you've got a 3D printer and maybe you just draw endometrium with glands in it and that dude just lays out the cells where they need to go? What do you think?
I think there's benefits to trying all avenues, right? I just don't know how far we will get with any one of them. So I think that's why we all need to be trying out different systems.
Yeah, you want me to try the 3D printing then? You want me to spend my money on the 3D printing?
No, and I think they're really important approaches to be using. I think, for me anyway, one of the important points is to not do stuff just because everyone's doing it or it's a cool thing. It's more about, will this help me answer my question? And what's the best way to do that? And I think sometimes, these like technologies like organoids say, there's a big leap and then it's a bit static for a little while and then there's going to be the next big jump to do like something we wouldn't have thought of we could do 5 years ago. So
Yeah, the endometrium's hard because it's so complicated, right? It's so complicated, different cell types. That's what I think we learned when we went to the single cell sequencing and stuff, and the spatial.
And there's stuff in there we don't even know about.
And we haven't even talked about endothelial cells, the environmental, the immune system cells in there, which are, the endometrium's highly vascularized.
Yeah, absolutely.
And those.
Oh no, I hold my hand up and say we haven't even we're looking at the very basic cellular composition of the endometrium, and
it's a heavy lift. Well, Niamh, you generate a lot of data, and I just want to remind you that the Journal of Dairy Science has a new author loyalty program. Are you aware of that?
I was not aware of
that. That is what you need to know these things, okay?
Yeah.
You probably, I don't know if you're a member of the American Dairy Science Association, but you need to be, okay? You need to be, because, once you've touched a dairy cow, you pretty much have to join. Okay?
Yes,
if you're a member of the American Dairy Science Association, let's remember that we have one of the sweetest price points for open access, gold open access publishing.
Good to know. Yep.
Much less expensive than like cell or developmental cell or something like that. And we have a new author loyalty program where if you publish with us and if you review with us, you get credits towards your article page charges.
Oh, that's really good.
Yeah, brand
awesome.
new.
Really
Brand
cool.
new.
Yeah.
You have to have an orchid ID.
Okay, I have an orchid ID.
Okay, you have how many?
Huh?
You have one?
I think so.
This is a running joke.
I hope so.
I hope so. There's more than one of me out
God,
there.
that's a frightening thought.
Yeah, that's a frightening thought. Anyway, so I'll bring that up. Yeah, so we're going to close right now, unless you have anything else you'd like to talk about. We've been talking for about an hour now. We always talk for an hour. I don't know how it all happens that way. But I want to ask you a few questions, because I bet you have some things to say here. Any favorite people, living or dead, that inspired you in your career? William Shakespeare? No?
I would be way more eloquent if that was the case. God, inspirational people. Oh, this is one of those questions where I'd be like, I should have said that.
I should have studied. Okay, I should have studied. Okay, give me another question. You ready?
Sure. We can circle back to that one.
We'll circle back to that one, okay. Career advice for young people. Like you got some new person, they're 18, they say, oh, Professor Forde, I want to be like you.
Again, frightening thought. Anyway, honestly, it's so cliched, but find something that does excite you and makes you enthusiastic, because that helps drive you when the times aren't so great, I would say.
Science is too hard for it not to be fun.
Yeah, exactly. I think so. And I think you need to have that passion and enthusiasm and interest to keep you going, I think.
You have to be curious, right?
Definitely.
It helps to be super curious.
My nickname as a child was How Come, because I used to just follow my mother around going, but how come? But
how come, right? Yeah. If you were not a scientist, pub singer in Galway, I don't know. Can you do Irish dance? That would be a good career.
Terrible.
No? Terrible?
Yeah.
Play the fiddle? No.
If I wasn't a scientist, I would probably be unemployed.
Unemployed?
Yeah.
Unemployed on the streets of Galway.
I honestly, I don't know. I would struggle to figure out what other job I could do or who would employ me. As I said at the start, I really like doing things that I really like, but I'm not great at doing things I don't.
You could be a podcast guest.
I could be.
You're good at it. You're good at it.
Thanks.
So you're going to give up on a favorite person? Nobody corny.
A favorite
Nobody
person?
corny.
Oh, that's a different question.
Okay, a favorite person.
Okay.
Living or dead?
One of my favorite people is Bill Bryson.
Bill Bryson? Bryson, he wrote the books on hiking or something?
Yep, he has this wonderful, so if people don't know who he is, he's an author from Des Moines, Iowa, and he wrote these wonderful travel books, and he also wrote a short history of nearly everything, and he just has that wonderful inquiring mind, and the way he gets across information is in a humorous and informative way, and I just think he's an awesome person, and I met him about 3 weeks in when I was into my job at Leeds. He happened to be doing a book tour and one of the stops was Harrogate in the north of Leeds. So I went and got my book signed and he was like, that doesn't sound like a Yorkshire accent. And I was like, no, and he's like, why are you here? And I was explaining like I've just started a job, and then he started talking to me about, is it true that women's eggs decline in terms of their fertility after the age of 34? And I was like, I can't believe I'm talking to Bill Bryson about oocyte developmental competency in Yorkshire. I think seems like a wonderful person in how he gets across. He's a scientist without being a scientist, if that makes sense.
I think he wrote something on hiking the Adirondack Trail.
He did the Appalachian Trail, and he's done loads of stuff like touring around Australia, the US, the UK and stuff, but some science books as well. Would recommend Bill Bryson.
And did you see the late toy show this year? I'm just wondering.
I didn't.
Oh,
no,
okay.
I'm sorry.
That's an aside. Okay.
Yeah.
Anyways, you'll catch it next year then.
Yep.
Well, Niamh, I knew it would be fun. I knew it would be super interesting. I knew you would be super great. Thank you for spending time with us. You feeling better today now?
Thanks for having me.
It's been awesome. So, all right, everybody, that brings us to the end of this episode of Dairy Digressions with Niamh Forde, Professor Niamh Forde from Leeds. Don't forget, if you enjoy Dairy Digressions, please like and subscribe. If you have any positive feedback for Niamh, or positive suggestions for me, if you have any speakers you'd like to tell us about, please tell us at adsa@adsa.org. Thank you again, Niamh.
Thanks a million, Matt.
This has been Matt Lucy, your host. Until next time, enjoy your science. Thank you very much.